So, okay, I don't know where my pointer is, but we'll manage. What? Okay. I hope so also. Okay, so previous chapter. Can you close the door there? Okay, so apart from the article that we talked about last time, we talked about started talking about molecular genetic mechanisms, and these are, it looks like a very general term, but generally when we talk about molecular genetic mechanisms, it's mechanisms of transcription and translation. And again, for someone that that has a background in biology, this is very basic, but we'll in the end of this class, in the end of this chapter, when we're going to talk about now, we'll talk about things that are not basic and are unique to neurons. Like in every chapter, we try to give the neuronal approach to that. So, we talked about the central dogma and went over the structure of nucleic acid. The dogma is like the DNA is transcribed to RNA, then translates to protein. But we saw that there's a lot of species of RNA that are non-coding. Okay, and non-coding, we mean non-coding to a protein. So only mRNA is the one that is responsible as transferring the information from the DNA to the level of the protein. But there are a lot of other types of proteins, of RNA, that are like tRNA, that is responsible for the translation itself in the RNA. And we also have, nowadays we know that huge areas of our genome are actually coding for genes, for RNA genes, that are not coding for a protein. Okay, so either short or long RNA, that are not coding for protein, but have function. We showed this schematic of how it happens in the cell, a little bit of reminder of the molecular structure of DNA, and then we showed the structure of the DNA double helix, and the pairing. We mentioned that the pairing between the bases is done by hydrogen bonds, and the stacking, the stabilization of this whole structure is due to the hydrophobic effect and van der Waals interactions between the I talked about this the stacking of the bases that have van der Waals interaction between them that stabilize and compress this structure of DNA. Another thing that's very basic to know about DNA is that G binds to C and T binds to A, and this is always the case, almost always the case. And the discovery of DNA, we said that there might be different forms of the structure of the DNA, but mostly B is the most common form. You have about 10.5 pairs per turn of the helix, of the double helix of the DNA. And that proteins that need to interact with the DNA often bind to the grooves of the DNA in order to identify the coiled structure of the DNA. We said that the DNA inside the cell is in a state that is filling the nucleus. It's not like if you might have seen like chromosomes in caricatures and stuff like that. So they're not in the form of compact chromosomes. All the chromosomes in our DNA are actually in like loose conformations, or they're filling the nucleus. But still, they're compact locally around special proteins that are called histones, which is these proteins that you see here. So they're coiled. The DNA is coiled around these histones, and they're very important to if you heard about epigenetics and the modulation of DNA and stuff like that. So a lot of it deals with these histones and the modification that they affect the transcription of the DNA and the expression of the DNA. We define what is a gene in general. So a gene can be either protein or RNA. That's very important to remember. Like here you can see it only in this gene is coding for a protein, but there can also be a gene that will stop like in this level and not be translated to a protein, but will still have function. So we still define it as a gene. We said there are different regions in the genomes, and what's important again to state is that all of these structures that you see here, the 5' unsaturated region, and again, this is a good place to talk about the directionality of the DNA. We always talk about directionality, okay? So although the DNA is always double-stranded, only one strand is defined as a gene. You can actually have on the same double strand, you can have two genes that are completely separate from one another, but one of them is in this direction and the other one is in the other direction. Okay? So this is the 5. Most of the time when you're going to see like by convention, when we show a sequence of DNA, the left side would always be the 5' or the 5 end, and the right side will always be the 3' unless it's stated differently in the specific case. But what's important to understand is that all the elements that you see here, all of them are sequences of DNA. These are not proteins, they are not something else. When you hear about promoters or enhancer silencer sequences and stuff like that, or 5' translated region, introns, exons, which we're going to discuss later, all of these elements are DNA sequences. Okay? So every gene, for example, has its own promoter. Sometimes this promoter can be very unique, and we use this promoter sequence often in molecular biology and also in molecular neuroscience to target specific genes or cause the expression of specific types of elements that we want. So we went over the steps, or briefly went over the steps of transcription, but basically this is a simplification just for you to see. The only thing that you see here is the RNA polymerase in the DNA strand, but there are a lot of different factors that are involved here, like you saw in the video that I showed you. But in general, again, it's important to understand like the way that the RNA polymerase interacts with the promoter regions, and from there it starts transcribing a single-stranded RNA on the basis of one of the strands of the DNA. So it's making a complementary. So we said that the base pairing can only be A and T and G and C. So in RNA, we don't have T, we have U. But basically it's the same, that you have a complementary strand that is being created on the basis of the DNA. And what's also important to remember is that the first part of the strand of the nascent or the new strand that is being created is always the 5'. So that's the first end that's coming out, and that will be important for processes that we're going to talk about later. So again, the directionality, and I showed you the movie. And then we talked about the structure that RNA can take, like, yeah?
Every time the 5' coming out, it's always translating transcribing on the third. I mean, from 3 to 5.
Yeah.
Does that mean that it's always going to be only, it means that there isn't like an option of expressing the other, it's like right, each gene has both 2 options, right? But each is in a person, right?
Each gene has 2 options? Like,
yeah. Like each, you inherit genes for each, whatever, for each.
Yeah, so you have, we didn't talk about genetics, or we won't talk actually about hereditary and genetics and stuff like that, but we have 2 copies. Each gene, you have 1 copy that you got from each parent.
So if it's always from 3 to 5, it means that you're always going to have a certain,
No, so the copies are double-stranded. It's not that one copy is like one strand and the other copy is the other strand. First of all, they're mixed, but regardless of that, you inherit from your parents the double strand. Okay, like so, from each parent you inherit a strand on its own. No, double strand. You inherit one double strand. Okay, DNA is always coming in the form of a double strand. When we say a DNA, unless I say like a single strand of DNA, I mean that the DNA is the entire thing, like the coiled structure. You inherit one set of these from each parent. So you can say that you have 4 strands. But that's important to. That's a good question. Also, it relates to what I said again, that the information of the gene is just coded in one strand. The other strand can be a totally different gene, unrelated to that gene at all. It's not like it's different versions of this gene. Like if I try to understand what gene is on the other side, if I read it like so.
It's not the same gene, a little bit of different mutations.
It's necessarily not the same gene, because it's a different sequence completely. Okay, like if you have this is the double strand, and you have A, T, like this is the sequence, and you have A, C, G, A, T, and this is the 5' end, and this is 3' end, and this is 5' end, and 3' end. So this can be a gene, okay? And when you try now to read the sequence, so if we read it together, the sequence of this gene is T, T, A, C, G, A, T. But this gene is A, T, G, C, A, T, C, G, T, A, A. It's completely different, okay? Necessarily completely different from the other one. It's actually, it's pretty amazing that the system is able to code 2 different genes on like the same sequence and get it like, it's insane, but still we see it. We see this phenomena happening. But although it's pretty rare, like most of the time, the other sequence or the other strand of DNA will not hold any significant information that we know. And also our genes are so, like we have a lot of genetic material relative to the amount of genes that we have. We don't have so many genes. Or the space that the genes themselves fill in the genome, it means that they're like it's pretty loose. So, but it does happen that the other strand will be a different gene. So so from each parent, so this is like, this can be the copy that we get from the mother, and we will get like the same, it can be the same or with slight variations, like genetic variations from the mother. So this is what we have this is what we mean when we have 2 copies of our DNA. Okay, so we talked about the structure of RNA. We said that RNA can also have like a secondary structure, which is secondary, we mean that is local folding. And this is because of the complementary, because we said that the RNA is always single-stranded. Okay, so actually only viruses have RNA that's double-stranded. All the RNA that we know of in the cell is in the form of single strand. So this single strand has a lot of base pairs that they can bind to each other, because they know how to bind through hydrogen bonds the same way that DNA strands, the 2 strands of DNA bind together. So over here you also will have bases that will be complementary to each other and can form this very intricate structure. For example, the tRNA, the transfer RNA, which couples between the mRNA, which is the messenger RNA that delivers the message of the gene to be translated. So it couples between the messenger RNA in one side through a region that's called a codon. Actually, this is the anticodon. And the other side, which will be the amino acid that is matching to this anticodon. Hopefully matching to this anticodon. Otherwise, you'll have big trouble. So we talked also a little bit about the ribosome and the mechanism. I showed you also in the movie. I think it's much better illustrated in the movie. about how translation occurs, which is translation of the information on the mRNA, and you need to understand how this information is presented. The biology knows how to read these triplets. Okay, so you can say that this is like a bit of information, this codon, and this codon translates to amino acid 4. And this codon translates to a specific a different amino acid, and the entities or the RNA that knows how to read these codons is again the tRNA, and it carries on one side the amino acid, and the other side it has the matching or complementary 3 bases that know how to read this codon, and this whole process is happening by the mediation, I don't know if you can say that, is mediated by the ribosome, by this whole huge complex that involves also more RNA molecules and 80 types of different proteins. Okay, this huge gray bulb that you see here. I mentioned in a few. You have a question? No. I mentioned in a few words the genetic code, which is again one of the, I think, the mysteries or one of the proofs that people use in order to say that all life originated from a single species, because all life have the same genetic code. Okay, all the organisms on the planet, with the exception of what I'm going to talk about in a few slides, or actually in next presentation, have the same genetic code. So it doesn't matter if you take an amoeba, a mushroom, a lettuce, or even a virus, anything. Every time you will see AUC, it will mean isoleucine. Okay, like the amino acid that will be like matched to this code will be isoleucine. Okay, so like this is like the lexicon or the deciphering, how do you say, the encryption code that is common to all biological things. So you can see why, maybe you can extrapolate already why people think that it's a proof that life started from a single origin, because a different organism could have different codons, because there's nothing special that connects isoleucine to AUC. I can tell you that. Because you have also AUU that's connected to isoleucine. They're actually sitting in different types. It's just the matching between them. But you can imagine that if you'll have a mutation along the way, for example, in one of these codons, or in the tRNA that matches this codon to threonine, then it will have very dramatic effects on the organism. Because now, the organism, every time that there was supposed to be a threonine, will have a like proline or something else, like a different amino acid in all the proteins. So that's a very dramatic change, and most likely will not survive. So this is why we think, like in evolutionary terms, that this is like changing the genetic code is something that is impossible in general in evolutionary terms, because even one shift, one deviation will have such a dramatic effect on the specific organism, where it happens that this organism will not survive because all the proteins, most of the proteins, like, will not be functional.
But don't different species have a preference for different codons that code the same amino acid?
Yeah, they do. They do because they can control for example, sometimes like because the ones that know how to decode these are the tRNAs. So you have like, for these 4 anticodons, you have 4 different tRNA genes. So there can be organisms that only have like these 2 tRNAs, and other organisms that only have that only express these 2 tRNAs. Or because they don't have like, for example, genes that have this anticodon. That could be in very simple organisms. But in most complex organisms, but still, they won't have a variation. Like any variation that they have, if it's significant, then if they only need these 2 codons, then there's no reason, like, if this mutated, it won't have any effect. But it's like a circular thing. If it's very important, and this is a very basic property, then if it's changed, then it will have dramatic effects. Okay. But there is one example that is different. So, but we'll talk about it in next slide. So, this is a nice animation of how this translation process occurs. I think we saw the movie of the Central. Did we see the movie of Central Dogma or just the translation movie? I think we just saw the translation movie. So I owe you a movie, and I don't want to. You should see the sequel. Okay. So this is something that really summarizes, I think, this chapter really nicely.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene.
These are the yellow things are single nucleotides. They are being linked together through a phosphodiester bond that you, for sure, if I wake you in the middle of the night, you know that this is the bond that makes that exists between subunits of DNA.
At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing.
We will talk about this, the splicing, like the process of splicing in future chapters, but obviously you understand up until what I told you now that it's much more complex than what we learned. It's even much more complex than what you see in this video.
Which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called the ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay.
Why do we have introns that they just get cut out?
We'll talk about it, but some of them are also functional, but they also allow the cell to, I'll just say a few words, that they allow the cell to, like for example, different cells in different tissues can express different proteins. Like let's say you have a cell in the digestive system that doesn't need all the elements in a specific protein, that then the specific cell in the digestive system can splice them out, and then you can get alternative splicing. So you can get from one gene different types of proteins according to there are actually genes that have insane amount of splicing in them. And this is actually one of the things that are very different, for example, between mammalians non-mammalians, but vertebrates and other organisms, is the amount of splicing that they have. For example, humans, we have probably the most alternative splicing, like the most variability in the amount of genes that you can make, the amount of proteins that you can make from a single gene in the genome. Okay, where am I? Yeah. So, questions on this chapter. Match each molecule to the molecular process it is directly performing in the cell using the following list. So, what are the functions of RNA? Is it decoding the nucleotide sequence? 2, part of the ribosome? Yes, this is ribosomal RNA. Information transfer? Not really, but it directly performs in the cell. Okay, so 4, part of an enzyme. This is a trick question for someone that is, if you are protein biased, then you will say that all the enzymes are protein, which is not true. Enzymes can also be RNA, so it's true. And information long-term storage. So it's 2 and 4. So it's part of the ribosome, and they are part of an enzyme. And I told you that, but okay. So mRNA is in charge of information transfer. DNA, so information long-term storage, right? And tRNA is the actual molecule that decodes the nucleotide sequence or the molecular or the genetic code. And yeah, that's it. Okay. So in the DNA molecule, yeah.
For organisms that do store their genetic material as RNA, what form of RNA is that?
So a lot of time, I didn't talk about these special cases, but in these cases, it can be, there are no rules. Okay, for these organisms, there are normally viruses, and they can have either even the bacteriophage, which is one of the most famous viruses, it can have inside that family, it can have double-stranded RNA, single-stranded RNA, single-stranded DNA, double-stranded DNA. So they're all the options. In viruses, they don't have, I think because they don't have really, they're not reproducing by their own, then it's much more flexible. But in general, in all...
You wouldn't even use the same sort of categories to deal with that RNA.
Yeah. It's different, just there. It's not... In all the organisms that have reproductive ability, I don't know of an example. There might be an example that only RNA exists there. I think that all of them have DNA, but I might be wrong. And okay, so in the DNA molecule, all the correct sentences. So the strand orientation is anti-parallel. Yes, the strand orientation is parallel. No. Covalent bonds hold the 2 strands together. No. Hydrogen bonds hold the 2 strands together. Okay, so again, these are the hydrogen bonds between the bases that we talked about. And hydrogen bonds are responsible for the coiling of the double strand. No. What is responsible for the coiling of the double strand? The hydrophobic effect and van der Waals interactions. Okay. No. Okay. Managed to cancel it. In which molecular property do DNA and RNA differ? In the composition of bases? How are they different? Yeah, RNA has U instead of the T in DNA. Chemical bond between subunits? No, it's still a phosphodiester bond. Resistance to degradation? Yes. RNA is much more susceptible to degradation than DNA. And chemical bond between strands? No, it's the same. Again, here, when we said about the RNA is single-stranded, so I mean like the local folding. So it's the same type of chemical bond. It's a hydrogen bond. And single or double-stranded form in eukaryote cells? Yes. And catalytic activity? Yes. DNA doesn't have catalytic, as far as we know. Okay. So basically, all of these. So bonus, it's not something special. from which codon will the ribosome start and finish the reading. So start codon is always AUG, and stop codons are UAA, UGA, and UAG. I will use this as a reminder again that the difference between translation and transcription. In transcription, we don't actually know in eukaryotes completely what causes the RNA polymerase to fall off the strand. There are certain hypotheses about that, but we don't know. But it happens sometime. But in the translation, from mRNA to a protein, we know exactly what are the stop codons and what are the start codons, and what happens in each of the processes. So, we didn't talk about specifically about neurons. So, what does Coca Cola have to do with neurons? What? Okay. No. But yeah, nice connotation. So, this is a map that shows all the places in the world that Coca Cola is distributed. Okay? So, apart from Cuba, Sudan, Iran, Burma, and North Korea, all the other places in the world have Coca Cola. So, as a company, what? They have. So it's so probably also Sudan already has Coca Cola, and Cuba also probably. Yeah. Yeah. Also Iran probably. So the entire world, but you get the point. So Coca Cola has to distribute their product around the world and get it like in an efficient way to all their customers. So the fable, or what I'm trying to get at here, is that obviously there's no sense in that Coca-Cola will only produce the finished product, okay, like the bottled drink. They will manufacture everything in the US and then ship it all over the world, right? It doesn't make any sense because it's very heavy. It will get ruined. It can't deal with different supply and demands. So there's no way that the production of the finished product will be only in the US, right? Like it makes no sense. So actually, the way that Coca-Cola solved this problem is that they send, first of all, blueprints and instructions of how to make their plants and how to make the beverage, the different types of beverages. They have like hundreds of types of different beverages. And the ingredient and concentrates that they ship to local agencies or local enterprises in the countries where they're distributed, and over there the local enterprises regulate the production according to supply and demand, like different, and the local transport of the finished product to the different regions. So up until now we talked about, so why am I saying that? Because if we try to compare it to a neuron, so the neuron has to, if the soma or the cell body of a typical neuron is 20 microns, then an axon of a neuron can get to up to 50,000 times more than the thickness the thickness of the cell body. Then it poses like it's putting us in a very uncomfortable position if we now think that all the proteins are made next to the nucleus and now have to be shipped to the axon or to the dendrite. Okay, and this is what people thought up until 10 years. Okay, up until 10 years ago, something like that all the proteins, and this is apart from the how do you say, like the conception that the RNA doesn't have any purpose besides transferring the information from the level of the DNA to the protein. So if it only transfers the mRNA, the information from the DNA to the protein, then it just goes out from the nucleus, which is somewhere around here. You see this? So the mRNA just goes around outside the nucleus, and the protein is being translated here, and then the protein is shipped to wherever it's needed. But again, if you compare it, and I think it's a very good comparison between the Coca-Cola distribution and the neuron. Actually, the neuron has to go much longer distances than one Coca-Cola needs to ship their product. And still, it has to maintain functional synapses that have to change and learn. I don't know if you learned about that, but they have to grow, they have to shrink, they have to get pruned, it has 10,000 synapses, and each synapse has different needs. Okay, so it's actually pretty amazing that up until 10 years ago, people thought, okay, it's okay, they ship proteins. Okay, so nowadays we know that this is not actually the case, and each year we understand a little bit more about the phenomenon that's called local translation. And local translation is actually the concept that describes that mRNA is actually shipped in its form, like, so you can think about it as the blueprints to make the Coca Cola are shipped in the mRNA form and stored for like medium, I wouldn't say long term, but for medium terms of time. It can be days or like weeks and even more, shipped to distal locations for the synapse, and over there in the synapse the cell has all the machinery, like it has the ribosomes, it has the amino acids, it has everything it needs over there to produce the proteins locally, okay, and when it's needed. This also can happen much faster. I don't know if you remember, but we saw that for a motor protein to carry a cargo of proteins from one side of the cell to the other, if it's a neuron, it can take days, this shipment. And we obviously know that learning and changes that happen in synapses don't take days. So the local translation actually allows not only for specific synapses to change and to produce proteins locally, but also for it to happen very fast. And how fast? This can happen in minutes. Okay, so once the synapse is activated, in like 30 seconds you can start having new proteins locally in that region. This is like the model, again, that is like describing, you see that each person draws the neurons like the message he wants to send. So for example, in this article from 2014, they show the neuron that is like very distant, you know, the cell body is very distant, and the synapses and dendrites are very so the RNA is actually shipped in the form of RNA granules, which are very compact and very resistant to degradation because of this compactness of them. And once there is a signal, like a action potential or an activation of a synapse, then this activation would cause like a transient disassembly. Then the granule can reform after the activation and locally produce proteins that are needed to change the synapse or to learn. Okay?
RNA is reused?
The RNA could be reused. Actually, what we know today is that the RNA granules is like RNA and ribosomes that are already bound to the RNA. So it's so fast that even the ribosome doesn't have to find the RNA. Everything is like, you can think of it as a very compact granule that everything is stuck there. Like the ribosome are not moving. Nothing is moving, it stays like that. And then the activation signal, by mechanisms that we still don't know, causes this granule to release, and the ribosome can immediately start producing new proteins. And then it's said that some of the mRNAs are being recycled and return to their dormant state. So, and this is something that obviously you can see why in neurons is extremely important. And this is something that is really, I'll use very bad terminology and translation from Hebrew. This field is in its diapers. Okay, so so just an example of like different genes that people have, different mRNA molecules that people have started to tag in order to discover which. It doesn't happen to all of the genes. It happens. We know now today that it probably happens to most of the genes, or most of the mRNAs are locally transcribed in neurons. They're also transcribed in the nucleus. I'm not saying that like near the nucleus, but there's different distributions. For example, the beta-actin, which is a very important cytoskeleton protein, you can see that it's very nicely distributed throughout the dendrites of the cell. This is like a neuron. And GAPDH, which is a protein that's needed for splicing, for example, the processes that are needed next to the nucleus, is not distributed. Most of it is around the area of the nucleus. And CAMK2A, which is another very important protein that is in charge of plasticity. A lot of processes of plasticity. It's also very abundant in the dendrites. And recently, in 2015, people have started to we have now the technology and the microscopy to tag single molecules of mRNA and to track them in real time and see like what they're doing. So this is like a movie from Park et al. This is a science paper from 2015. This is for those of you who know, then this is a two-photon microscope movie, and the dots that you see here. I really hate this. Okay, so the dots that you see moving here are actually, this is happening in real time, and this is the shipment of RNA molecules, in this case beta-actin. As you can see that some of the molecules are mobile, are like moving, and some of the molecules are just located in spines, in synapses, just waiting to be translated. Okay. So again, you can understand that if the first time that we saw it properly in neurons is in 2015, then there's a lot to do in this subject, and obviously for neurodegeneration and all this stuff. There's a lot of work to be done in this field. So we're done with this chapter. And the next chapter I'll just start with the introduction of this chapter, which is biomembranes and cell architecture. Is again pretty basic, that it's very important for the next chapter, which will be transport of ions across the cell membrane, which is something that is very crucial, obviously, for neurons, like the ionic composition and all the transport of ions and neurotransmitters and stuff like that is a very key feature of neurons. So in order to understand this, we need to first understand what is the biomembrane, and what does it do, and how does it look in the cell. So in general, this is like an illustration of a very simplified cells. The cells of eukaryotic organisms, which we are part of, are divided into, they have internal organelles, and we define them as organelles because they have a membrane as a barrier. Okay, so the cell is actually a cell because it has this membrane. So it has a double layer membrane. We'll zoom into it in a second. But just to get you acquainted with the terms, is that we say that the internal space inside these organelles, for example, the Golgi, lysosomes, nucleus that has it has two layers of a double membrane. So the internal area, or the internal space inside the organelles, is termed lumen. The space that is inside the cell but not inside an organelle is called either the cytosol or the cytoplasma. And the space that's outside the cell is called the extracellular space or the exoplasma. Okay, and actually here the membranes are the ones that define. You can think of them as the skin or something of the cell. So yeah, I think we'll take a break, and then we'll start the chapter.
very different names. I've heard mattresses, which they need mattresses up there. They really, really do. It's a very long walk to go to the bottom of the lake. Sleeping there. Yeah, I need to have the money for it here too.
Well, I did learn everything over
I think I very much deserve it because I did the things for not have teachers. I mean, a little bit. No, no. The problem is any of those have to do just say, like everything in the this is like wage actually kind of wage hang out in the US in your farms. Really? Like so you're on the nutrition thing.
כאילו
I
איפה?
didn't realize it was
אני עכשיו מרגיש כמו הכי זקן בעולם. לא, האמת שאני לא... כן, כן, כן. זאת אומרת שהוא יזכור את זה.
It's very hard to do it.
זהו, שאלתי שאם אתה לא עושה את זה כנראה שכאילו לא...
Yeah, I agree. He's entertaining.
אבל
It's true, but he's like more of a cartoon character. No, I've noticed that they'll look at it. No. They'll look at it. I've always associated Donald Duck. My children with it. Personality, they're very specific. Whenever he gets angry, Donald Trump gets very Oh, did you agree?
I think you're like insulting
Donald Duck?
Donald Duck.
Okay. No, I agree. I just mean like they
Yeah,
very
he's
similar.
like a child.
Okay.
He's like a little kid. Does anyone want? I'm not being like very good at this.
What are the tricks?
It's like a, it's a banana monster, but it's kind of tasty. And it looks like.
Like quinoa?
No, I'm being honest.
She's being very nice, but she's standing here while we eat it. Do you want to like get rid of it in a way? So like you need the nutrients.
I was explaining why I'm not enthusiastically offering it, but you're
Okay.
welcome to have some.
I guess like you're vegan for most of the day.
It's a banana muffin.
Well, sometimes like the the day. No, if you're like
No. Do you want one?
here's the reward.
Again, like don't get your hopes up.
לא, בגלל שאנחנו לא יודעים לאט, אנחנו רב מול כאילו...
Okay. So, lipid composition and structure organization. So, I told you in the past that so we already saw in the past, but now we're gonna dive a little bit deeper into that, to the structure of the double layer membrane of the cell. So this is not an illustration. This is an illustration, but here you can see an electron microscope image of a cell, and here you can clearly see the two leaflets or the membrane bilayer that bound the cell itself from the cytosol, which is the internal part, to the exterior part or the extracellular space. And molecularly, it looks something like this. So we said that you have that the fatty acids in the cell are molecules that we said that are amphipathic. They have one side that is polar, which is the polar head, and they have hydrophobic tails that can either be 2 or 3 strands of these hydrophobic tails. And what is the type of chemical interaction that holds this structure together? Hydrophobic effect and van der Waals interaction between these tails to each other. Okay, so this is what holds them like this, and what causes them to cluster together is the hydrophobic effect. So hydrophobic van der Waals interactions, and what it actually does is that it makes it creates an impermeable barrier to molecules that are water soluble. Okay, so molecules that interact with water, or hydrophilic molecules, in order to cross this barrier, they will have to leave their water behind and cross this barrier, which is a very hard thing to do without any transport. But luckily in cells we have a lot of different molecules and different proteins that specifically know how to transport molecules that are needed across this barrier. But in general, molecules that are hydrophobic and small will be able to cross this barrier freely, and molecules that are hydrophilic and interact with water will not be able to cross this barrier. But we're going to talk about that in depth in next chapter. So the fluid mosaic model is what we biologists call the membrane itself. And the reason that we say fluid mosaic, first fluid, because you might think that this structure is rigid, but it's actually moving all the time. So the fatty acids are actually not bound to anything. There's no covalent interactions between them. So they can actually freely float around and move around this structure. And also many of the proteins that are not bound to anything, to the skeleton of the cell, or the skeletal proteins of the cell, can also move in a movement that we call lateral movement. So we say lateral movement because it's like moving along this structure. So again, you need to imagine and to use your imagination to imagine that these molecule or phospholipids can freely move sideways. So it's like a fluid, okay? And mosaic, because you will never see, like the membrane is not only phospholipids. It has a lot of different types of phospholipids, for example. It also incorporates cholesterol molecules, that we'll talk in a second what they actually do here, and also a lot of proteins and channels that are located inside inside the membrane. So obviously the composition of the lipids and the composition of the other elements that are inside each membrane will contribute to the physical properties of the membrane. And we'll talk about it in a second. So the steroid cholesterol, for example, which similarly to phospholipids, has a part that's hydrophilic, or the hydroxyl group here is hydrophilic, and these rings here, or the carbon rings structure over here, is hydrophobic. So it will always have the orientation that is something like this, that this OH group will point towards the hydrophilic part, and this group will interact with the tails of the phospholipids. Questions?
The proteins in the membrane, if it's like molecularly, they change their location, or it's just like moving
They could otherwise if they're not anchored. Okay, so but we're going to show in a second the different type of interaction of protein with the membrane. So first, regarding the composition and structural organization, if you have, for example, we're returning now to one of the first lessons that we talked about, which is the structure of fatty acids. So I mentioned like that we have unsaturated fatty acids like this oleic acid that has this kink, in contrast to saturated fatty acids, which like in this example. So if you have fatty acid like phospholipids that are saturated or that are made of saturated fatty acids will have a more rigid consistency, because again, what we said, that the van der Waals interaction will be stronger because they have more surface contact between one another. And if you have like a membrane that's more that has more unsaturated fatty acid, it will be more fluid. Okay, like it will have like a fluid-like consistency and not a gel-like consistency. And obviously, the lipid composition will affect also the thickness and the curvature. For example, phosphatidylcholine is a phospholipid that has linear that is linear in shape, or cylindrical in shape, and it will be mostly located like in areas that are straight in the membrane. But in areas where you have curvature in the membrane, a lot of time you will find phospholipids that have like a conical shape, like phosphatidylethanolamine in this example here. So, and the thickness is also different, and here comes also one of the roles of cholesterol. Cholesterol is not only bad; it can also be good under certain amounts. For example, cholesterol is one of the ways of the body to regulate the fluidity of the membrane. For example, a membrane, actually cholesterol works like a buffer in a lot of cases, meaning that if you will add the cholesterol to a membrane or to a membrane that's composed of a lot of phosphatidylcholine molecules or phospholipids, it will cause them to straighten out a little bit, and thus increase not only the thickness of the membrane, but it will also cause the membrane to be a little bit more rigid. Okay? What?
What causes that?
The cholesterol. Like the interaction with the cholesterol creates, again, van der Waals interaction that will straighten out this molecule a little bit and cause the membrane to be a little bit more rigid. Okay, for example, a lot of like animals that have to change the temperature that they live in will often have like more and less cholesterol in the membranes according to the time of year, for example, because the temperature also requires like a certain enforces a certain consistency of the membrane. And it can actually work in the other way around, although it's not shown here, which means that if the membrane is very structured, like in this case, adding cholesterol can actually make it more fluid-like, because it will like cause the fatty acids to interact less with one another. So that's why I say that sometimes in the body, under normal conditions, not in conditions that you eat McDonald's all day, but if you maintain a good amount of cholesterol, then it can act as a buffer of the cell, like making the membrane consistency. If it has to be more rigid, then it will always drive it towards the direction that the cell needs the membrane to be, like the consistency that the membrane should be. Okay, that's why I say that it's like a buffer, because it's
And also make it more rigid and also make it more fluid?
yeah. So it depends on the composition of the phospholipid, which it can also change during the life of the cell and during certain conditions of the cell. But for example, in also temperatures, for example, if the cell is exposed to lower temperatures, then the phospholipids will move less, and then the membrane will be more rigid, and then adding cholesterol will make it more fluidic, for example. So, so cholesterol can be a good thing, but also too much cholesterol can have bad effects. So, you can actually find different compositions of lipids in the outer leaflet and in the internal leaflet, and this is because the different environments that these leaflets are exposed to. Obviously, like the external leaflet is exposed to extracellular proteins, enzymes, and other factors, and the leaflet that is inside the cell is exposed to enzymes, proteins that are inside the cell, which are different. So these enzymes, for example, can cleave different regions or different bonds in the phospholipid. And also one of the reasons that we see asymmetry between the two leaflets is the way that these are synthesized in the cytosol of the ER, or the exoplasm of the Golgi. So the way that they're synthesized in the cell is also in a non-symmetrical manner, and this is why we see different composition of leaflets, of phospholipids in the outer leaflet and in the inner leaflet. There are also enzymes that know how to change the two, which can do this motion that we call the flip-flop, in which you can have like these two can switch between one another. But normally this will only happen in the presence of this enzyme. It won't happen like naturally, this movement. Okay, so the way that proteins interact with, we can categorize or divide the way that proteins interact with membrane into three categories. First, we have peripheral proteins that normally don't interact directly with the membrane, but are but are around the membrane and interact with proteins or with the cytoskeleton of the cell or the extracellular matrix to interact with protein that will then interact with the membrane specifically. So these are peripheral proteins. Then we have integral proteins that will span the membrane. Okay, a lot of time, for example, all the channels, ion channels, any type of protein that has to transport, or neurotransmitters will often activate channels that will allow the permeability or the entrance of ions to cells, will be integral proteins. And we can also find lipid anchored proteins which have this hydrophobic tail that just anchors itself to the membrane itself, and then they can move along these sides. They can also have this lateral motion, but they're anchored to one side of the membrane. Question? Okay, so integral or transmembrane proteins, which are the ones that interest us the most because they're a lot of the ion channels and other receptors are integral transmembrane proteins, will often have alpha helices or structures of alpha helix that will span the region of the membrane itself. Okay, so in this example, it's a type of bacterial rhodopsin, like a molecule that knows how to absorb light or to react with light. So it will have this, but this is a very common feature that we find in almost, I can say I know one example to a structure or to molecular structure that has beta sheets here instead of alpha helixes. Like most of the time, or the majority of times, you will have alpha helixes that will span the area of the membrane, and these alpha helixes will be hydrophobic or hydrophilic. They will be hydrophobic, and sometimes they will be half hydrophobic, half hydrophilic, because they will create a hydrophilic core in the center, and they will face outwards, they will be hydrophobic, and inwards they will be hydrophilic. And again, I return you to why the alpha helix is such a good structure to span the membrane, because by itself it doesn't have any free hydrogen bonds, and thus is not hydrophilic or hydrophobic by nature, and only the amino acids determine this property of the alpha helix. So we can also find different types of lipid anchored proteins that are most of the time will be covalently linked to these anchors. And again, this thing, the composition of these and the different types of proteins will often be found in the external part relative to the internal part. For example, this structure is sugars. I don't know if it's or hydrocarbons. It's very common to see outside the cell and not really inside the cell, like the anchors of the sugar anchor, which is a lot of time used as a signaling, as a signal of the cell to the health of the cell, to the things that the cell need. It often uses these hydrocarbons or sugar as like signaling molecules. And this is why they will be in the external part of the cell. Okay, so very briefly, we'll go over the organelles of the eukaryotic cells, which are very important for you to know. First, so this is like a very simple illustration of a cell. Obviously, this is not a neuron, and it has also implications of why it's not a neuron. But we're going to talk specifically on what happens in neurons in terms of organelles. So first of all, and what we're talking about today is the plasma membrane, which is what encapsulates the cell itself and defines the cell itself. Then you have the mitochondria, which is in charge, which is like the power plant of the cell. It's surrounded by a double membrane, actually, and its role is to take glucose and oxygen and fatty acids and convert it to ATP. So this is like the organelle that is in charge of production of. You can say why do we compare it to a power plant? So it's like a power plant can take like energy from the sun, from coal, from any other thing, and convert it to usable energy, to available energy. So like we have electricity that is available energy, the cell has ATP. So there's a. Now I need to shut off the microphone, because what I'm going to tell you is a theory that I came up with, and it is very controversial, and I can't. So one of the things that we have to say about the mitochondria is that it's a very weird organ. Okay, like we think that along evolution, some prokaryotic or some type of bacteria somehow created this symbiotic relationship between eukaryotic cells and the mitochondria itself. So the mitochondria itself doesn't have a lot of DNA, but it has still DNA of its own, and it produces also proteins of its own. So, but when the cell replicates, it also the mitochondria is also replicated. So, and the information for a lot of the genes that the mitochondria needs is inside our genome. So in terms, we're inseparable. We're like the same organism, the mitochondria and the eukaryotic cells, but still we always think of the mitochondria as something that a little bit strange is happening there. So do you remember that I told you that the genetic code is something that is one of the proofs that we are all from the same origin, right? So, and there is just one example that I know. Actually, there are two, but the most interesting example of where there is deviation of the genetic code is in the mitochondria, actually. So in the proteins of the mitochondria, we can see in a few amino acids, I don't really remember which ones, that a deviation from the genetic code, which is pretty, like, for me, and now I have to say that I'm not serious. Okay, that's why I also turned off the microphone. It's just for comic reasons. So I don't know if some of you know the hypothesis of midi-chlorians, or the story of midi-chlorians in Star Wars. But midi-chlorians were actually, they were thought of, and also George Lucas admits that the idea was taken from mitochondria. That these are like so that these are like the power plants. So when you think about it, it's kind of a cool idea to think about an extraterrestrial being that somehow incorporated with us, and this is what supplies us with the force. Okay? Because we all know that the proportion and the concentration of midi-chlorians is equivalent to the amount of force that you have in. For example, who had the most midi-chlorians in history? You know? Who?
Anakin?
I think Luke Skywalker, no?
That was Anakin's father.
Never mind. Anyway, so we know that, for example, the Jedi Knights have a high concentration of midi-chlorians. So actually, I always thought about it as a cool idea, but then when you combine this fact with the fact that they have a different genetic code, I don't know. It starts to make sense. So now I can turn on the microphone again.
So, is your theory that if we actually find someone with, I guess, more mitochondria, they may actually be able to semi-use the force?
Maybe. No, but this is Anyway, it's an interesting thing. So, lysosomes. Lysosomes are organelles that are related. They have an acidic lumen, so the lumen of the lysosomes is actually much more acidic, which means it has a lower pH than the physiological pH. All the other organelles in general have physiological pH, which is around 7.2 and 7.4. But the lumen of the lysosomes have more acidic pH, and the reason for that is that we think about the lysosomes as organelles that know how to recycle like wasted organelles, and also peptides or small or proteins that either come from outside the cell, or also proteins and peptides that are left over from the degradation that we talked about with the ubiquitin process. Okay, so for example, the small peptides that are produced from the ubiquitin process, they could be shipped into the lysosomes, where they will be completely degraded, some of them to single amino acids, some of them to more basic molecules, to be recycled by the cell. You have the next organelle is the nuclear envelope, which is a double membrane. Okay, and when I say double membrane, don't get confused with every membrane has two leaflets. So the nuclear envelope is what encloses the genetic materials, and you can also see these spots, which are the pores. These are like the openings from which the mRNA or other molecules that have to exit or enter the nucleus go through. So this is like the portal to the nucleus. The nucleolus is actually not encapsulated by a membrane. It's just what we defined as a region where we can find the RNA, which is needed to. RNA is again the RNA that is part of the ribosome, and the nucleus is nucleus itself, which is filled with what we call chromatin. Okay, and again in these terms there's a lot of mix-up. Okay, when we say chromosomes we mean like specific subdivisions of the DNA, but chromatin is a word to say the entire genetic material. So all the chromosomes are the chromatin. Okay, and chromatin specifically is also the combination of the DNA and the proteins that wind it or interact with it, like the histones that we saw. And it's stupid to say it's the site of mRNA and tRNA synthesis because it's obvious. So the smooth endoplasmic reticulum, or in short, smooth ER, is where lipids are synthesized. So I'm not sure you can see it, but it's like this structure or this structure over here. It's where lipids are synthesized, and also there's some kind of a detoxification. I don't know, detoxing things. And the rough endoplasmic reticulum is actually, I didn't tell you because we didn't go really in depth in the translation process. But for example, if you have a protein or a protein that has to be integrated inside the membrane, this integration usually happens during the translation of the protein itself. For example, if you have an mRNA that is coding for a protein that has to be integrated or incorporated into the membrane, then when the first amino acids will start coming out from the ribosomes, they will normally carry a signal that the cell will know that it has to take this whole complex, which is the ribosomes and the mRNA, and direct it to this structure, which is called the rough ER. And you see these spots over here. These are actually representing ribosomes, and this is why it's called rough. So the ribosomes are not previously bound to the rough ER, but after the peptide, after the first few amino acids start coming out, there are special proteins that know how to identify the first amino acids, and they can take this whole complex, stick it to the structure, which is the rough ER, and the rest of the translation will happen inside the ER. So it will be translated into the lumen of the ER, the protein. And after that, it will be normally processed in an organ that is called the Golgi. that we always say it's like the mail or the post office of the cell that is in charge both of finally processing like cells that have to be, like, for example, transported outside of the cell, and also membrane proteins that have been synthesized in the rough ER. For example, remember what I told you before, like one of the reasons that people thought that there's no local translation of mRNA is that they didn't find rough ERs in the synapse. Okay, they said that because the rough ER is only around the nucleus, then it doesn't make sense that to have local translation, because you know that a lot of the proteins that have to be incorporated in the membrane have to pass through the rough ER in order to be incorporated to the membrane. So, but nowadays we actually know that in synapses you have everything. Apart from the nucleus, you have everything. You have Golgi, you have rough ER, you have the mitochondrions, and you have everything that is needed in order to both make membranal proteins and also proteins that are not membranal. And secretory vesicles are vesicles that normally carry proteins or other components. Nowadays we know that they also carry RNAs and other type of molecules that are meant to be secreted outside of the cell. Okay, so specific organelles that are unique to neurons, there are actually two of these. You can actually divide them into more, but in general, the first is the synapse, which is the area of contact or the relay of the chemical message or electrical message between two neurons, where you have a lot of secretory vesicles, for example, that carry neurotransmitter that will fuse with the membrane of the cell and release the content into the extracellular space, and then they will be recepted by the target cells. And in the nervous system, we also find the myelin sheath, which is we can also say that it's a type of organelle. It's not an organelle of the neuron. It's an organelle of a type of cell that's called the Schwann cells, or also oligodendrocytes can have them. But they form these loops, these conductive or isolating loops around the axons of the cell, which really increase the efficiency of transporting the signal from the cell body along the axon to long distances. You learned about that, right? You learned about that? Yeah. Okay. So the synapse is a tight structure that's formed from a contact between two neurons. I really like this image because it really shows you the difference between a caricature, how a synapse looks, and how a synapse would look in reality. So this is like an EM micrograph or an EM image of a synapse. This is actually highlighted like the synapse itself, so it will be easier to see. And as you can see, you also have like huge mitochondria that can be really inside the synapse itself. And I don't know, I'm not sure if you can see, but you have a lot of secretory vesicles over here, and this is like the synaptic cleft, or the area of contact between the two cells. Myelin sheath, like I told you, it facilitates the conduction between nerve cells and manages to, and because of the myelin sheath, the nerve cells can conduct or can relay electrical signals to very, very long distances. And lastly, the neuroligin neuroexin complex is what holds the synapses together. So again, and this is something again that's very unique to neurons. The neuroligin neuroexin complex is actually composed of two proteins. One of them is postsynaptic, which is neuroligin, and the other one, which is neuroexin, is presynaptic. And these two proteins are what we call scaffold proteins. These are the proteins that actually hold the synapse together. And it's actually very interesting to find that we can find different types of neuroligin and neuroexins in different types of synapses, which again, you would not think that this is the case, because you say, okay, the type of synapse, why would it matter what type of protein holds the synapse? But we actually find beta-neurexin and neuroligin 1 in excitatory synapses that have glutamate, and in inhibitory synapses, for example, we'll find mostly neuroligin 2 and either beta-neurexin or alpha-neurexin in inhibitory synapses. And I can tell you that it goes much more specific than that. We have nowadays we know of at least 5 types of neuroligens and another 4 types of neurexins, and the combination between them really probably defines specific types of synapses. And for example, a lot of autism cases are linked to mutation in the neuroligin gene, because probably there is like malassembly or malrecognition of synapses because of these mutations. So again, it's very important to me in this class to show you the difference between cartoon and reality. So this is how synapses look in cartoons. This is my favorite. This is actually like most inaccurate, which is like the force, which obviously there's no mitochondria here, so it doesn't make any sense. But this is actually how it looks in reality. So each one of these arrows, so this is like taking from a brain, and this is an EM image. So first of all, you can see that everything is filled with material. It's not like it's isolated and you have like a synapse in space. So everything is filled with cells and biological materials. This, for example, is a myelin sheath or a bundle of axons. This is like a side section, and each one of these arrows is directing you to the synaptic cleft. So this is an axon, this is a dendrite, this is a dendrite and an axon, and you can see it for yourself. So it's like it's like a mess that's going on here, but luckily biology sees logic in that, which helps us function. So how do these organelles look in EM? This is the mitochondrion. What is this? This is very easy. The nucleus. What is this? Nuclear membrane. These, you actually don't know, it's secretory vesicles. And here it's very hard to see. So these are like Golgi vesicles, the endoplasmic reticulum, plasma membrane of the cell, and the intracellular space, which is very rare. Most of the time, everything is filled with cells. More examples. This is just glycogen because it depends from which organ it's taking, but never mind. These are more images of mitochondrions, or this is peroxisomes that know how to special organelles that know how to break down again proteins. This is the examples of smooth ER, and this is rough ER. So again, you can see these spots are ribosomes that are actually in this moment translating their proteins into the lumen of the smooth ER. What are these? Do you know? First of all, this is the nucleus. Yeah, this is the nucleolus, and this is what we call heterochromatin. Okay, so this is a good place to talk about a little bit about different densities of DNA. So I told you that the chromatin is a name that we use to describe the combination of DNA and proteins. And I don't want you to think, we said that the DNA is filling the nucleus, but not all the DNA is in the same state. Some of the DNA is in a compact state, and some of the DNA is in more loose state. And again, you can imagine, and it makes sense to, this is also what we see, is that in certain, like the areas that are more loose, are the areas of DNA that have genes that this cell specifically needs. So because this area of DNA is more loose, then the factors that need to bind to it and transcribe the genes that the cell needs have an easier job, and it's more accessible. And portions of the DNA that we can say that the cell maybe doesn't need are compact in these granules or these clusters, which we call heterochromatin. Okay, the other part of the chromatin, I didn't write it here, but it's called euchromatin, the more loose parts of the DNA. Okay, for example, in different types of cells, not all the cells, and actually I think it's pretty true to say that no cell, no cell expresses all the genes. Okay, each cell only expresses a certain set of genes that's needed, because we have 20,000 genes, protein coding genes in our genome. You'll not find a cell that expresses all of them, okay? Because the cell has a specific function, and for the function of that cell, it expresses the specific protein that it needs, or the specific genes that it needs. So the other part of the material, the other part of the DNA, a lot of time is kept in a compact manner and is not transcribed at all in the form of heterochromatin. This is a zoom in into the mitochondrion. Again, you can see here really nicely the inner membrane and the outer membrane, and these crested or these clefts of internal membrane that are inside the mitochondrion. Okay, so questions about this part? No? Yeah. So the cytoskeleton, we talked a little bit about it when we described motor proteins, because they walk on the cytoskeleton. But we know that the cytoskeleton is composed of three types of filament. The actin filament, which is again what you see here, that the subunit is just made of a lot of actin proteins. Again, all of these are proteins, and they're very small in diameter. And I don't know if you remember, but these are not like the main highways of the cell, but these are like the small. They make up the small alleys or alleyways of the cell. The microtubules, which are the larger structures, they're 24 nanometers in diameter, and they're made of alpha and beta tubulin, like I showed you in previous classes. And the last category, it's much less defined, which we call intermediate filaments, which are about 10 nanometer in diameter, and their subunits are very variable, and they're made from a diverse family of proteins. So, in general, we can categorize it into actin filaments, intermediate filaments, which are much less defined, and microtubules, which are the main structure components and highways of the cell. For example, the cytoskeleton is actually what keeps the cell, because I told you that the membrane itself is not something, is like a fluid. Okay, so the thing that gives the cell its volume is actually the cytoskeleton, and the cytoskeleton also interacts with the membrane itself through what we saw, like peripheral proteins and other types of membrane proteins. And for example, in this nice image of the cell culture, you can see that the microtubules, they also have a directionality. In all the cells, you have a center that's called the organism center, or the microtubule organizing center, which is a specific spot in the cell. It's not necessarily near the nucleus at all. It can be in a specific location of the cell, and this is the place where the microtubules originate from. Because I told you that the microtubules, they also have directionality. They have a plus side and a minus side, and the place where they start, or the start location, is in this structure that's called the microtubule organizing center, or centrosome. And in this nice image, you can see that the microtubules are marked in green and the centrosomes are marked in yellow. In neurons, we can also, if we stain for cytoskeletons, we also get these really nice images. And for example, in this primary hippocampal neuron, which is again, you don't see any other neurons around it because it's like isolated in the petri dish. But in this primary hippocampal neuron, the DNA or the nucleus is stained with a blue dye. The microtubule associated protein, or MAP2, which is a microtubule protein, is stained in green, like you can see here. And the actin filaments, which again you remember that we said that they're more local, and a lot of time are like the alleyways that are springing from the main highways, or to the very rich in dendrites, are labeled in red in this case. So each one of these are synapses. Each one of these red dots are synapses that are enriched with actin filaments. Questions? Okay, so I don't know if I'll finish the purification of cells and their parts, but similarly to what I told you about the proteins, that in order to study the proteins we need to isolate the proteins, the same thing goes to the organelles in the cells. Okay, so start from purification of cells. One of the most common ways nowadays to identify a cell or a type of cell that you're interested is to tag it with a fluorescent protein. Okay, so for example, in this case, this is an image I took, by the way, like a few years ago. So this is primary culture. So this is on a petri dish, and all the cells that you see here are neurons. And neurons that we say are cholinergic neurons are neurons that express the components of the cholinergic system are tagged with a red fluorescent protein. So the way that we did it is actually that we didn't do it, but other companies do it, is that if you want to tag specifically this type of cell, you can take what we call a marker gene, or for example, in this case, one of the enzymes that is responsible to synthesize acetylcholine, which is called CHAT, or choline acetyltransferase. We can take its promoter, or the region of DNA that is before this gene, and instead of putting the CHAT gene, the coding region for the protein itself, we put a GFP protein. Okay, so now each time that the cell is intending on producing choline acetyltransferase, which for us means that this cell is cholinergic, because only cholinergic cells express this protein. So instead of producing this protein, it will actually produce a fluorescent protein, which in this case is TG tomato, which will help us identify it with a fluorescent microscope. So after we identified the cell, we can actually isolate it with a very cool device that's called a fax. Not fax like a fax machine, but fluorescent activated cell sorter. And it has the ability to separate cells according to their fluorescent properties. So it looks, it's actually a very simple concept, but it does it really efficiently, is that you have a laser that detects the fluorescence of the cells,